Thank you for your presentation, Ava. It is now time for the Q&A. To ask Ava a question, just type it in where it says your question here and then press submit. So our first question, Ava, and this one asks, did the authors look at T-cell exhaustion after antigen tumor encounter? And were the T-cells containing the inducible on switch exhausted more or faster than the WT CAR T-cells?
Thank you. Great question. Yes, we have examined exhaustion both in vitro and in vivo, and we found that activating the activation switch, turning on the activation switch, reduced exhaustion and kept these cells and vivo with tumor. The groups of mice that have the activation switch turned on by immediate treatment can eliminate these tumors, even really big, high numbers of tumor cells, while the CAR T cells alone, with vehicle treatment, no activation switch, cannot do that. This work has not been published yet, but hopefully it's coming.
Excellent. Thanks, Ava. Our next question asks, the persistence of CAR T-cell therapeutics is a determinant of long-term success in patients. Did you try any repetitive stimulation of your cells following the on-off switch with fresh tumor cells in vitro or in vivo?
Yes. I guess I kind of answered this question already in my answer to the first question, that we did indeed try it both in vitro and in vivo, and in vivo we went out to about 150 days, and the T cells are still going strong. They can handle new tumor challenges, and they don't seem to be exhausted. The only issue that might come up that we need to address is when tumors have this enormous challenge from the immune cells. They undergo immune splitting, and they can reduce the antigen on their surface and escape the immune cells by reducing the antigen. These cells are specific for the antigen, so if they don't see the antigen, that will affect their effectiveness, tumor killing ability. So we are looking into ways to handle that problem.
Excellent. Thanks, Ava. And the next question asks, I suppose sort of following on to that, given that rapamycin is an immunosuppressant, is there any worry that RAP might impede other aspects of a patient's immune system outside of the CAR T-cells, and how would this impact the sort of therapeutic outcome?
Yes, it seems to me from the questions that there's a little bit of confusion with the rapamycin and the rimidacid, and I do understand that this is a complicated system. Rapamycin was used as an agent to turn on the safety switch, so we are using it to reduce or eliminate our activated T cells. So rapamycin being an immunosuppressant is actually helpful in this situation, because we want to dampen the immune response since we are having an overactivated immune response. It would be counterproductive for the immune response to elicit tumors, so that's why we need to balance these treatments carefully. I hope this explains it better.
Excellent. Thanks, Ava. Our next question asks, can specificity to this would be lost when you're expanding?
We have not seen that. Our constructs are stably transduced. The T cells are stably transduced, so we have not seen the construct being lost in the T cells. Of course, the T cells are not 100% transduced. We will have non-transduced cells, some of it, or cells that only have part of the constructs, since these constructs are on different plasmids, the safety switch and the activation switch and the CAR. So that will happen. We are in the dual switch product. We do a selection process to try to enrich for those cells that have our constructs. But still, as you can see from the in vivo data, we don't see that the T cells would lose the specificity against the tumor.
Excellent. Thanks, Ava. And just a quick reminder to our live audience, you still have time to ask a question. To do so, all you need to do is type it in where it says type your question here and then press submit. So our next question, Ava, and this one asks, this individual was wondering how you engineer the T cells to express the CAR and switch genes.
Yes, so we use retroviruses, retroviral constructs to use our T cells. Some other companies, as I mentioned in the beginning, use lentiviral transduction or Sleeping Beauty transplants, and other companies use the talons or the CRISPRs. For us, retroviruses seem to work. They give us a stable transduction, and it doesn't seem to have an adverse effect on the cells.
Excellent. Thanks, Ava. And our next question asks, does IMC work independently of tumor antigen activation?
I'm not sure what the question is aiming for. If you're asking if IMC activation alone could drive tumor killing, then the answer would be no, because the tumor killing is antigen specific. So you do need the CAR for that. The IMC is driving the survival and proliferation and cytokine production of the cells. So as I mentioned, it works more like a co-stimulator. If you would just transduce a mix of T cells with the safety switch itself, and if there be clones of T cells in this mix that would be reactive to the tumor, then of course it would work. But you do need that receptor that recognizes tumor antigen for the killing actually to take place.
Excellent. Thanks, Ava. Our next question asks, did luciferase transduction of CAR T have any effects on persistence or functionality? Is that something you can answer?
We have not done any experiments directly to address that question. It is a valid question. It's kind of a technical challenge. So if you don't have the firefly luciferase or the vanilla luciferase in your cells, then it's harder to identify them. As I mentioned, there are ways, the vector copier and the flow cytometry. That would be a quite big undertaking looking to have a high number of animals where we would have to take samples and analyze them side by side with the in vivo imaging. We didn't see the need for that so far. There wasn't any obvious detriment to the cells. As I mentioned, we had experiments where the experiment run out to 150 days, and the mice were healthy. The T cells looked healthy. They were proliferating and present by in vivo imaging. So I am not concerned about this. But thank you for the question.
Excellent. Thanks, Eva. Our next question asks, if you sort of re-challenge with the tumor after elimination of the tumor and safety switch induction through that, so will you have re-eliminated the tumor? Sorry, is that something you can help answer?
Yeah, sure. So
rephrase the question slightly better than I did.
Yes, I'm trying to unpack that. So after we use the safety switch and re-challenge the tumor, if we keep giving remedicide to activate the activation switch, and there is the tumor antigen from the re-challenge, again, also depends on how much drug we use to activate the safety switch. If you remember, I showed the dose response for the safety switch. We can give a lot of drug and eliminate almost all the T cells, or we can just give less drug and eliminate most of the T cells. So if we have remaining T cells that have the CAR, that have the activation switch, and we use remedicide to activate that activation switch, then those T cells will expand and will control the tumor as much as they can. Obviously, it's a little bit of a numbers game if we can totally eliminate the tumor, but it is possible.
Excellent. Thanks, Eva. And just a quick reminder to our live audience, you still have time to ask a question. To ask one, all you need to do is just type it in where it says type your question here and then press submit. So our next question, Eva, and this one asks, is there a specific type of solid tumour to which this therapy is particularly effective towards?
Yes, it's a very good question, very important question. Obviously, the solid tumors that have some kind of tumor antigen that is specific for the tumor, ideally not expressed on healthy cells or not a lot of healthy cells and very low levels, that would be ideal, because if we don't have that, then we have a hard time balancing out the T cells, making sure they attack the tumor and not the healthy tissue. So obviously everybody is looking for those targets on tumors, and PSCA that we use is one of them. HER2 is not that ideal. HER2 is unfortunately quite widespread in the body at low levels, and so that is definitely a limitation of the CAR T therapy, and probably that's one of the reasons why a lot of companies are looking into NK cells now, because NK cells don't need that antigen target. They can have a CAR, and a lot of companies are adding that too into the NK cells. But yes, this is definitely a new area to work on.
Excellent. Thanks, Ava. Our next question asks, can there be a memory T cell for a tumour?
Yes, there's memory T cells, and they can provide long-lived immunity. We are trying to characterize ourselves and see if we have those. Activating this switch, the activation switch, definitely makes our cells long-lived, and whether they can be characterized memory T cells or they are just long-lived effector T cells. That's still a question that we haven't don't have a satisfactory answer to, but we are working on it.
Excellent. Thanks, Ava. And our next question asks, do you personally think your inducible CAR could be used for targets that could also be found in the T cells?
Very good question. It doesn't really matter what I personally think. I can tell you that we have worked on targets like that, just like other companies like CD123 and CD33, and it's definitely a challenge. There's a process called fratricide where the T cells would eliminate other T cells that have the same antigen, the CD123 or the CD33 on them. So as I mentioned, we have been working on it. We had some progress, but there's still more to do in that regard. It is a challenge.
Excellent. Thanks, Ava. Well, I'm afraid that is all we have for today. I'd now like to thank Ava Morsha for today's presentation and for answering your questions. I would also like to thank the webcast sponsors, Merck and Alma, and you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you join us again soon.